The hormone rewrite when male fish grow eggs. Rivers remember. They remember every molecule that enters them, every discharge, every trace compound, every chemical fingerprint of modern life. For centuries, Britain's rivers have carried the byproducts of civilization, industry, agriculture, waste, filtered, diluted, released, and forgotten. And yet, in the early 1990s, scientists who looked beneath the surface of these familiar waters uncovered a truth so unexpected it forced an entire field of science to rethink what pollution truly means. Because something in the rivers was changing fish, not killing them, not leaving them visibly diseased, but altering them at the most fundamental level of biology. Male fish were found with egg cells embedded within their testes. Egg cells oocytes growing where sperm-producing tissue should be. Intersex conditions had been recorded in wildlife before, but only as rare curiosities, isolated anomalies. This was different. This was widespread. This was repeated. This was impossible to ignore. These were not laboratory specimens. This was not an artificial experiment or a genetic oddity. These were wild fish caught from ordinary English rivers, including long stretches of the Thames. What was once considered an anomaly became a pattern. As sample after sample was examined, the scale of the phenomenon emerged with unsettling clarity. In some locations, the majority of male fish showed feminized characteristic egg cells replacing sperm, gonads no longer conforming to biological norms. This was not a disease. It was not a parasite. It was something in the water. When researchers mapped where these intersex fish were found, a striking geographic pattern appeared. Upstream, intersex was rare. Downstream, it increased, and below sewage treatment works, it surged. The closer fish lived to treated wastewater discharges, the more severe the feminization became, rivers receiving higher proportions. Of effluent, particularly during dry seasons when dilution was limited, showed dramatically elevated rates of intersex. This was not confined to a single river. The River Thames, the River Trent, the River Ira, the River Avon. Across England, the same relationship appeared again and again. The key factor was not dramatic chemical spills or obvious industrial contamination alone, but something far more mundane. The proportion of treated sewage flowing through the river. In some waterways, especially during low-flow periods, treated wastewater made up a significant fraction of the river itself, unknown to many. Every toilet flush ultimately finds its way back to the river. The river was no longer just habitat, it had become a delivery system. The river itself had become a living biological experiment. Then came another clue invisible to the naked eye, but unmistakable to biologists. Male fish downstream of sewage treatment works were producing vitellogenin. Vitellogenin is a protein essential for egg yolk formation. It is normally produced only by females under the precise control of estrogen. Males do not need it. Males should not produce it. And yet there it was circulating in male fish at levels that sometimes rivaled those of spawning females. Vitellogenin became a molecular warning signal. The fish were not simply damaged, they were responding hormonally to a signal in their environment. Hormones govern development, growth, reproduction, metabolism, and behavior. They operate at concentrations so small that traditional toxicology once dismissed them as irrelevant. Trillionths of a gram can determine sex, fertility, growth, and behavior. Some chemicals interfere with this system. They are known as endocrine disrupting chemicals, EDCs. They do not poison in the traditional sense. They do not burn tissue or shut down organs. They whisper instead of shout. They send false biological signals mimicking natural hormones, blocking receptors, or altering hormone synthesis and breakdown. They only need to arrive at the wrong time, especially during development. Sex determination in fish, as in humans, is not governed by genes alone. It is hormonal. Timing matters. Concentration matters. Exposure windows matter. And among all the endocrine disruptors detected in rivers, one compound stood apart. 17-ethinylestradiol, known simply as E2. A synthetic estrogen designed to be potent, designed to resist breakdown. designed to work at extraordinarily low doses. E2 is the active ingredient in many oral contraceptive pills, a medical breakthrough that transformed reproductive freedom worldwide. But once the pill has done its job, E2 does not simply disappear. It is excreted in urine and feces, often as inactive conjugates. In sewer systems and sewage treatment works, bacteria can convert these compounds back into biologically active estrogen. Conventional sewage treatment was never designed to remove hormones, so E2 flows onward diluted, treated, but still biologically active into rivers. At concentrations measured in parts per trillion, E2 has been shown to induce vitellogenin production, feminize male fish, and disrupt sexual development. Laboratory studies confirmed what field observations suggested. Exposure to E2 at environmentally realistic concentrations caused male fish to develop intersex gonads, produce egg proteins instead of sperm, and lose reproductive capacity. This was not speculation. It was experimentally reproducible causality. Feminization is not merely a curiosity of anatomy. When males produce egg proteins instead of sperm, fertility declines. When gonads develop abnormally, reproductive success drops. Intersex males may produce fewer sperm or none at all, leading to reduced fertilization rates. Over time, entire populations can collapse not because fish are dying, but because they are failing to reproduce. In experimental settings, long-term exposure to E2 has led to complete population failure within just a few generations. In the wild, the effects are slower, quieter, but potentially just as devastating. Fish are sentinels. They reveal dangers before humans notice them. Humans share the same endocrine architecture, the same hormone receptors, the same developmental pathways shaped by hundreds of millions of years of evolution. While drinking water is treated more rigorously, exposure to endocrine-disrupting chemicals occurs through many routes: water, food, plastics, pharmaceuticals. The concern is not immediate toxicity. It is subtle, cumulative disruption, especially during early life. Human endocrine systems are most vulnerable during specific windows: fetal development, infancy, puberty. During these periods, hormones guide organ formation, brain development, and reproductive maturation. Disruption does not always cause immediate illness. Instead, it can alter life trajectories, fertility potential, hormone sensitivity, disease risk later in life. Scientists continue to investigate links between endocrine disruptors and declining sperm counts observed globally, the altered timing of puberty, reproductive disorders, hormone-sensitive cancers. These links are complex, multifactorial, still unfolding, but fish provided a crucial insight. Hormonal pollution does not need high doses to matter. If synthetic hormones can reshape wildlife at vanishingly small concentrations, what else might they be influencing silently within us? The quiet consequence. This is not a story of villains. It is a story of unintended consequences, of chemistry colliding with biology, of systems built to manage waste never designed to manage hormones. The issue is not the value of modern medicine, but its journey beyond the body. The story is no longer about discovery. It is about response. Today, advanced sewage treatments such as activated carbon and ozonation can remove E2, but they are not yet universal. Rivers do not accuse, they do not warn. They simply reflect what we release into them, and in their currents they carry the biological signatures of our choices long after we've forgotten making them. The feminization of fish taught science a new language once spoken, not in death, but in subtle biological change. It reminds us that the most powerful pollutants may be those we never meant to release at all, and that sometimes the future of human health can be glimpsed quietly in the anatomy of a fish. If you found this exploration meaningful, subscribe to the channel to ensure you don't miss future stories uncovering the hidden consequences of our modern world.